This method can help resolve major challenges in pancreatic islet transplantation, such as immune rejection, post-transplantation islet revascularization, and survival. The main advantage of this technique is that this easy-to-adopt approach enables surface engineering of living cells. By reshaping the cellular landscape of pancreatic islet, it will improve graft function, survival, and ultimately the therapeutic efficacy of islet transplantation. The implications of this technique extend towards cell-based therapies because the therapeutic outcome of cell-based therapies is also limited by low cell retention and poor survival. Demonstrating this procedure is Jingyi Yang, a graduate student from my laboratory.
First, weigh out 0.69 grams of heparin and dissolve it in 2 milliliters of ice-cold deionized water in an Eppendorf tube. Dissolve 0.23 grams of NHS in 0.5 milliliters of ice-cold deionized water in an Eppendorf tube. Then, dissolve 0.77 grams of EDC in 0.5 milliliters of ice-cold deionized water in a 50 milliliter conical tube. Mix the NHS and EDC solutions in a 50 milliliter conical tube. After leaving the mixed solution at room temperature for 30 minutes, centrifuge it at 12,000 RPM for 10 minutes to remove excess EDC and NHS. Following this, add 30 milliliters of cold ethanol to the solution and centrifuge at 12,000 RPM for 10 minutes. Now, add 1 milliliter of 10% StarPEG amide 8 to a 50 milliliter conical tube. Then, add 0.67 milliliters